Bueno, pues nada, pues muchas gracias a la conexión por permitirme o por invitarme a hacer esta presentación, sobre todo porque yo no soy del CSIC, soy del IMDEA Nutrición. Para los que no sepáis, pues los IMDEA son como una especie de CSIC que tiene la Comunidad de Madrid. Y bueno, pues, pero sin embargo, pues como sabía que existía este hub de microbioma, pues me pedí entrar pues la verdad es que me aceptaron y me parece muy interesante toda esta iniciativa. Voy a hablar, bueno, estoy a cargo de, como decía Julia, del grupo de Quimioinformática e Inteligencia Artificial de este INDEA, de INDEA Nutrición. Estamos en Madrid, en el campus de la Autónoma, y mi charla va sobre una plataforma computacional para el diseño de fármacos dirigidos al sistema microbioma intestinal huésped humanos. Primero de todo, los agradecimientos, antes de que me quede sin tiempo, pues a la izquierda son toda la gente que ha contribuido a este trabajo, especialmente Andrés Sánchez Ruiz y María Estigarraga, y a la derecha todas las agencias financiadoras. Bueno, el área de investigación de mi grupo es química informática e inteligencia artificial de compuestos bioactivos. Estamos interesados en moléculas como los compuestos de alimentos, los metabolitos microbianos, toxinas animales, como fuente de espacios químicos nuevos para el diseño de fármacos. Utilizamos una combinación de química informática, química computacional, datos experimentales en base de datos y machine learning, data mining para modelar la asociación entre las actividades biológicas de estas moléculas, sus propiedades fisioquímicas y su estructura. Y luego desarrollamos modelos generativos de inteligencia artificial, de deep learning, para diseñar nuevos compuestos. Hoy, pues como voy a hablar de los metabolitos microbianos y bueno, pues aquí no hace falta que incida mucho en este foro, pues en la importancia de la microbioma, especialmente la microbioma intestinal en la salud humana. Entonces, pues esta microbioma interacciona la señalización entre esta microbioma y el huésped se da a través principalmente de una serie de moléculas pequeñas, de metabolitos microbianos, que interaccionan con proteínas tanto del huésped como las proteínas bacterianas. Y se ha visto, la investigación en microbioma, pues indica que la desregulación de estas interacciones, porque por ejemplo un metabolito se sobreproduce, etcétera, pues puede ser una causa de distintas enfermedades en áreas tan diferentes como el inflammatory bowel disease, el cáncer de colon, enfermedades cardiovasculares, etcétera. Entonces, a partir de aquí ha salido una nueva, digamos, modalidad de diseño de fármacos que son los metabolitos, o sea, las moléculas miméticas de metabolitos microbianos dirigidos a este sistema, ¿no? El sistema microbioma intestinal huésped. Es muy interesante porque puede dar, por una parte, lugar a nuevos quimiotipos para desarrollar fármacos nuevos y, por otra parte, para expandir el espacio de dianas al microbioma, ¿no? O sea, sería pues expandir no solamente tener como dianas el proteoma humano, sino también el proteoma bacteriano, que serían como dos órdenes de magnitud más. Entonces, bueno, pues estos son unas revisiones y papers en este área recientes que pues indican es un área que está cogiendo mucho fuerza y distintas empresas farmacéuticas, por ejemplo, están revisadas aquí en este paper, pues están utilizando, digamos, o están metiéndose en este campo nuevo. A partir de aquí voy a hablar en inglés porque la presentación está en inglés y me resulta más fácil hablar en inglés. So our group started a project in this area. That is, the aim of this project is develop a platform, a computational platform for the design of human microbial metabolite mimetic as drugs. So since we are a computational group, we use a combination of cheminformatics, bioinformatics, and artificial intelligence. So the idea is to first, starting with the with the compounds, the microbial metabolites, using cheminformatics, perform a physiochemical, structural biodistribution characterization of these molecules. On the other hand, using database searches and virtual screening, identify all the compound target interactions of these molecules with both microbial and host proteins, and using bioinformatics, estimate the disease relevance of these interactions. And finally, using artificial intelligence, design methods to generate molecules, synthetic molecules for targeting this system. So we started with this initial cheminformatic characterization of the molecules and perform an analysis of the microbial metabolites in terms of their physiochemical and structural properties. Here I show some examples of microbial metabolites, and then we ask ourselves what characteristic or what features do these molecules have in terms of their physiochemical and structural properties compared to the oral systemic drugs typical so far for drug discovery. And if we were able to identify different compound subsets within them. So as model system, we use the metabolites in the gut compartment present in the human metabolome database. Here in this database, you can find molecules that are only present in the gut, which in turn, a big subset of them, which we call gut FL, are derived from fatty lipids, but there is a smaller subset of molecules only present in the gut, but without or not derived from fatty lipids. This is about 700 molecules, and we call them gut no-FL molecules. On the other hand, you have molecules that are present both in gut and in serum. This is about 500 molecules. And for comparison purposes, we use the subset of the drug bank comprising oral systemic molecules. And then we perform a systematic analysis for statistical patterns in of these molecules, of these three compound sets in different areas. So we search for similarity to drug-bound molecules. We search for scaffold distribution of in the molecules, ionization classes, physiochemical properties, chemical classes, etc. And from here, to cut a long story short, what we were able is to derive a set of rules for medicinal chemistry. The idea is that you can, if a medicinal chemist wants to synthesize a molecule based on these three subsets of these three groups of molecules, is able to use these rules to generate molecules mimicking each of the three of them. For instance, the gut-FL molecules are big, lipophilic, flexible molecules with no rings. They are mostly neutral, and they are gut lingerers. They remain in the gut and are unable to cross the gut wall. But on the other hand, you have the other two groups, the gut serum subset and the gut no-FL subset, are molecules that are small and polar, while in the case of gut serum molecules, they are more similar to drugs. They have more aromatic and heterocyclic scaffold, as it is the main ionization class, and they have higher hydrogen bond acceptor and donor content. They are gut traversers. They are able to cross the gut wall. While in the case of the gut no-FL molecules, they are gut lingerers. They remain in the gut, and they have more aliphatic and pure carbon scaffold, and they are mostly neutral, with lower hydrogen bond acceptor and donor content. An interesting property of these molecules could be, if you want to design a molecule targeting this system, is to design the molecule so that it would remain in the gut. In this way, if your molecule remains in the gut, you are going to expect less secondary effects, less toxicity, etc., because the molecule will not locally and not go to the system circulations. So in order to do this, or to help in the design of molecules targeting and remaining in the gut, we derive a model to predict gut permanence from molecular structure. So starting from the four compound sets, we merge into a gut lingerers compound set, the two compound sets that remain in the gut, and merge the two compound sets that are able to cross the gut wall. We merge them into a gut traverses compound set. And then we develop a machine learning model of a super learner type. This is basically an ensemble model that uses many different machine learning based models to improve the prediction and the generalizability capabilities of the model. And from the statistics of the prediction for the external test sets for both fatty lipids and no fatty lipids, you see that statistics are pretty good, so, which give us confidence in the reliability of these predictions of this model. And here in this repository, you can find the, the GitHub repository where you can get the model. Then we moved to the second phase of the project, which was the full characterization of the target space of these molecules. So we asked ourselves if we could, I mean, how much do we know about the chemobiological space of these molecules? So if we plot in a matrix like this, in the y-axis, the microbial metabolites, and the x-axis, the human microbial targets, which of these cells contain one or more published interaction? And if we were able to expand this knowledge with computational predictions. So we devised this analysis and prediction pipeline, starting from the gut metabolites on the human metabolome database. We use chemogenomic databases, specifically we use BindingDB and Campbell, to find all the published interaction in this for these molecules. We found around 2,000 published interaction, which we later analyze in terms of chemical classes versus target classes, etc., and scaffolds. And on the other hand, we use this tool, similarity ensemble approach, which is a cheminformatic tool to predict targets, to predict proteins interacting with molecules from the molecular structure of the compound. So in this way, we are able to fourfold expand the number of interactions with predictions. We were able to validate retrospectively 127 interactions, and using bioinformatic tools, we were able to prioritize a subset of these proteins. So more specifically, this is the distribution of all the published interactions for both human targets and microbial targets in terms of chemical classes and target classes. So you see that we have around 451 targets for human targets and 56 microbial targets. After the predictions, so you compare this previous heat map with this other, you can see that many cells that were previously empty, now they have one or more predicted interactions. So you can see that now we have more than twice expanded the number of human targets to about 1,000 human targets. And in the case of microbial targets, we have now 180 targets. So we have provided all these predictions as data sets openly. So the idea is that with this, by releasing these predictions, we expect that people across the world can test these hypotheses and confirm them, so that we hopefully will be able to accelerate the knowledge in the target space of microbial metabolites. As I said before, we were able to validate a subset of the predictions. In particular, 127 of these interactions we were able to validate through a retrospective analysis. We realized that the C method uses a snapshot of Campbell 27, that is 3 years ahead of the database that we use to find the published interaction, which is Campbell 33. So in this way, we were able to see if the predictions based on Campbell 27, some of them were actually published in Campbell release 33. And as a matter of fact, as I said, we found 127 of these interactions that were previously predicted and in Campbell 33 had appeared as published. And you can see here the distribution of the Tanimoto coefficient, which is a measure of the similarity of the predicted compounds compared to the compounds that are known for the corresponding targets. So you see that some of the predictions have similarities as low as 0.3, meaning that they are not just trivial prediction of very similar compounds, but they are confirmed predictions for compounds relatively dissimilar to the known ones. So this opens the opportunities for finding new chemical novelty for these proteins. And then later we use bioinformatic tools to prioritize the proteins obtained from the previous analysis. So for the human proteins, we use the Pharos tool. Pharos is a very interesting tool that aggregates a lot of information from GWAS analysis, gene expression analysis, protein expression, tissue distribution, etc., and also interactions with known compounds. So Pharos is like a tool that is able to provide for each protein in the human proteome a sort of validation score for some disease. Okay, so what we did was to identify in Pharos the human proteins statistically associated with intestinal disease as a surrogate of the biological relevance for the gut microbiome-host system. From here, we identify this set of significantly associated proteins, and we cross this database, these proteins, with the proteins that we have obtained for human in the previous analysis. And by crossing this information, we get around 500 high priority host targets. So we expect that these proteins are very high or strongly associated with the gut host system, microbiome host system. On the other hand, for microbial proteins, these are not represented in Pharos. So in this case, we use an essentiality score system, which is the GIPTOP 2.0 tool that tells you if a protein is essential or not. So from here, we identify a subset of 63 essential proteins that should be considered as high priority for microbial targets. We went further in this analysis of the target space of microbial metabolites, and then we asked if we could get information about at an atomic level, how are these interactions in terms of the functional groups and types of interactions that do these molecules establish. So here is a secondary bile acid, which is a very typical microbial metabolite. And in this, for all these molecules, we ask ourselves what functional groups are characteristic of the microbial metabolites, what types of interactions do they establish with proteins, and how are the functional groups used for these interactions. So we devised this other analysis pipeline, starting from the 4 compound sets that we have mentioned before. We identify all the protein complexes containing any of these molecules and found a total of 22,000 PDB structures. And for each of these molecules, what we did was to analyze the functional group of the ligand, the interaction that they establish with the protein in terms of hydrogen bonds, PP interactions, salt bridges, etc., and cross these two informations to find patterns statistically interesting. Here I show the distribution of functional groups. You can see that from this distribution, it can be seen that gut serum subset is the most similar to drug bank, while gut is the most dissimilar to the drug bank. So this confirms our previous analysis from physiochemical and structural properties. Here I show the distribution of the usage of the different interaction types, like hydrogen bonds, halogen bonds, PP interactions, in terms of for the 4 composites in the analysis, the drug band, the gut serum, the gut neutrophil, the gut effect. And surrounded by a box are the type of interaction or the compound class that is overrepresented for this type of interaction. For instance, gut serum uses an overrepresented set of hydrogen bonds, while for instance, drug band makes a high usage of cation P interaction, etc. And from here, you can see the association between the functional groups from the molecules with the interaction types. So you see, in general, that the same functional group uses the same interaction patterns for the different compound sets, but in some cases, the same functional group is used or uses different interaction patterns depending on the compound set. Again, to cut a long story short, from here we have learned a lot of ways that these three molecules establish interaction with proteins. So we have learned that these molecules have different ways to interact with proteins, which in turn are different to the way that typical oral systemic drugs interact with proteins. So it allows to develop new ways to interact with proteins that can be used in the molecular design of molecules based on these compound sets. Now we are in the last phase of the project. We have performed a lot of structure-based virtual screening, and we are applying generative artificial intelligence to develop molecular designs for these methods for these molecules. So as conclusions, we have studies we have found specific physicochemical and structural profiles for these three compound sets that have provided rules for medicinal chemists to design molecules based on them. We have developed a superlearner model to predict gut permanence from molecular structure. We have characterized the target space of these molecules and we have expanded the target space with validated prediction that can be tested through focus screens. The three metabolite sets display alternative modes for protein binding divergent from those of drugs. And we are developing generative artificial intelligence models to design molecules based on all these conclusions and data sets, and we are currently developing them. These are all the papers we have published so far in this area with all the repositories and data sets. You can expand everything about all the details there. This is related information. We have performed similar analysis for food compounds. Y eso es todo. Sí, pues muchas gracias por vuestra atención y si tenéis cualquier pregunta estaré contento de responderla.